The second is, if you want to entice them once they're there, don't tell them until you're halfway into your talk that's actually about Drosophila. And the third is don't use whimsical titles. It's serious science, right? But perhaps whimsical titles fit a whimsical question, which drives the talk today. Why do fruit flies live so darn long? It's a whimsical question, it's a silly question. It's a silly question, of course, because we all know that Drosophila is such a popular organism for the study of aging because it's so darn short-lived, and that's why we can do interesting things like selection experiments. And it's a silly question because of the fact that we've done so much experimentation with Drosophila. We know all about how this organism ages, right? More than any other organism. We know about the evolution of aging in Drosophila. So we can test some ideas, some framework laid out mathematically very nicely by William Hamilton that explains some ideas that have been laid out before Hamilton himself. Those ideas specifically are that mutations can accumulate later in life because of that declining force of natural selection for continued survival. They're relatively unchecked. So Woody Guthrie had half a dozen kids before he was diagnosed with Huntington's chorea and died of early death. Now the other side of that is that an individual may benefit from higher early life activity even if it comes at the expense of later activity. And some speculated that Huntington's chorea may even have those kinds of antagonistic pleiotropic effects early in life in humans. Okay, so those are two ideas about mechanisms for the evolution of aging. Those ideas have been well studied in this model system for Drosophila melanogaster. So here's an example of a kind of experimental approach, one that was applied by Michael Rose, by Leo Luckenbill, and by several others since. And that is selection by modifying what you might call the Hamiltonian cliff, the force of natural selection. It's a very simple experiment. If you look at that force of natural selection for continued survival in a short-lived stock maintained on, say, a 2-week generation time, you can see that the force of natural selection declines abruptly from 100%, at least in females, to 0 over a very short space of time when the organisms are turned over in lab culture, discrete generation culture. If you extend the force of natural selection for survival to later ages, as Rose did and Luckenbill did in the early 80s, you have the Hamiltonian cliff much later in life. You select for individuals that can survive beyond that point in time. It's a simple experiment, and it's generated some very nice evidence for antagonistic pleiotropy, evidence for mutation accumulation in other experiments, and you can see very readily that selection experiment has modified the longevities of these populations of baseline and old, and old type Drosophila. So just as an approximation, if you look at sort of the LD50, and it correlates very nicely with the mean longevities here, you've got close to a doubling in lifespan induced by this simple manipulation of the timing of reproduction. Okay, so that's a nice demonstration that those kinds of mechanisms may apply in the evolution of aging in that Hamiltonian framework. My question today is, why do these organisms, these baseline populations which reproduce this early, survive till ages of 4, 5, 6 weeks in typical laboratory cultures? Now you'll notice that this work was done in the early 90s when I was a graduate student with Michael Rose. At the time, I mused upon this problem, and I realized that Michael had mused upon the problem a few years earlier than this, about a decade earlier, as had Brian Charlesworth, his PhD supervisor. And so it comes to me another decade later to start thinking about this problem with my graduate student, Chris Kimber, who asks the same question. The darn animals are still living 5 weeks. So these are the stars of the show today, these unsung heroes, the baseline populations. These are the IVES population collected by PTIs in Massachusetts, descendant populations called B. They're maintained on a simple 2-week life cycle where they lay eggs on the 14th day /0 day, and around they go in their vials back to go to the starting line again. Okay, so they're discrete generations. They reproduce at about 4 or 5 days of age, and they've been selected relentlessly this way for over 800 generations. Now you might think that would select for characteristics that would confer seminal parity upon these flies. A burst of reproduction followed by relatively rapid mortality. So is that in fact happening? Well, I told you in 1993 they were living about 5 weeks on average. A decade before that, they were living about 5 weeks. A decade after that, they're living about 5 weeks. And Chris Kimber, my graduate student who started working on questions related to sex and aging, showed that they're still living about that length of time today in the laboratory. So it struck us that it was kind of an interesting question, that despite relentless selection, and despite the clear predictions of the evolutionary theory of aging, that mutations should accumulate to reduce lifespan, it didn't seem to be going down, at least not a whole lot. That mutations should accumulate, or that antagonistic pleiotropic alleles should eventually accrue in these populations. In other words, that lifespan should be going down, at least a little bit. Well, is it? The first thing we did was asked if the longevity had in fact changed, and what could we do to look at this question. Longevity assays, depending upon exact conditions, can give you somewhat different answers even using the same stock populations. So this is a bit of a rough analysis, but I'm going to use it to motivate some real experimental work later on. So we dug into the data, we found that there's heaps of it. Rose has done tons of experiments involving longevity. So many, in fact, and it's various academic offspring, including myself. So many, in fact, that we could pick and choose studies which had very similar environmental treatments, very similar mating status treatments. We threw out all the data with virgins, for example. We didn't use any that were used as measurements of stress tolerance, all those kinds of things. We took them out. We took experiments we thought were largely comparable. And we looked at a transect over 30 years of data. And what we found was that, true to our suspicions, there really was no hint of a decrease in longevity over this time period. These transects, these regressions are pretty much flatline for both females and males. So our perception seemed right, at least within this noisy ability to detect. We could not detect a decline in lifespan in these populations, IV and B. Okay, but we wanted to take it a step further and ask a more significant question, and that is, if they're not declining, why? Okay, so it could be, as Charles Ruth and Rose mused, that there's been an exhaustion of antagonistic pleiotropic variation in the populations. That exhaustion must have occurred really early in the domestication process, because the longevities now are comparable to those measured in the early 1980s. Okay, 30 years. It could be that drift is an exceptionally weak process. Populations are maintained with an E, in the range of 10 to the 3, and perhaps there just isn't a significant accumulation of mutations. So that's a viable possibility, and we can't exclude that entirely, but I'll show you that we can perhaps counter that possibility somewhat with the data that we have. So in 1997, Michael and I wrote a paper, and we talked about what we called the pleiotropic echo. And that's the idea that maybe there were some positive pleiotropic interactions early in life that led to extended lifespan. But it was just musings in another paper where we noted this phenomenon of exceptionally long life in stocks that have been selected for very early reproduction. So Chris and I wanted to look at this more carefully and actually do some manipulative experiments. The first thing we wanted to do, though, was to look at the relationship between longevity and early life fitness of adults in these populations, or actually specifically in the Ives population. That's the only population that we actually did this in. So we want to look at the standing genetic variation. Now, if these populations are at some kind of quasi-equilibrium, we might expect a positive relationship between early fitness and longevity by virtue of mutations selection balance in those populations. We would expect a positive correlation, but perhaps a weakly positive correlation. But if we experimentally enrich those populations with new mutations fluxed in through a mutation accumulation protocol, we'd expect that to be a more positive and stronger positive relationship. Okay, so we use the system I inherited from Bill Rice, that hemiclone system. I won't bore you with the details of it right now, but what it essentially allows us to do is to take something over 99% of the genome, the big chromosomes 1, 2, and 3, lock them together as a linkage group, as if they were a single gamete, and actually propagate that gamete in large numbers as sperm through males, sans recombination. Okay, so here's our hemiclone, as we call it, We can make lots of them, that's what we usually do, but in this case, we instituted an experimental protocol for mutation accumulation by transmitting a single spermatozoan from one generation to the next via males in our mutation accumulation lines. Any mutations that were acquired in that generation were passed to all descendants. At the same time, we kept the same hemiclone in larger numbers in a population, so we wouldn't have the same kind of mutation accumulation process occurring. Natural and sexual selection were still going on. And so we tested these animals for controls or for MA hemiclones for total fitness and longevity. Okay, so what about the standing variation first? The standing variation showed what we basically expected for the relationship between fitness and longevity in females, a weakly positive association between female fitness and longevity. Males were not quite so persuasive because we have a lot of variation in the measurement of male fitness in the assay. It's non-significantly different from zero. So the females seem to support us and the males are equivocal. It's not surprising that the mutation accumulation process led to a decline in fitness in both females and in males, and confirming some of our own results from mutation accumulation assays earlier, confirming some work from Anil Agarwal's lab and others. We see a stronger impact of mutation on male fitness than female fitness. Males are the more sensitive sex, it seems. And we see also a decline in longevity, a significant decline in both sexes. Now, when we look at the genetic correlation between the sexes, we find that in tune with our predictions, it became a stronger and more positive association, and we see a steeper slope and a tighter relationship between early life fitness and longevity. Okay, why is that interesting? Well, it's interesting because it suggests that mutations that decrease longevity are naturally occurring in these populations. When they decrease longevity, they have a substantial impact upon early life fitness, reproductive success, that is, both female and male. So that suggests that selection is actually actively maintaining this surplus longevity that we see in these populations, the 5 weeks instead of the 5 days of longevity that we might have expected. It suggests that there are bands of positive pleiotropy between these early life acting genes that affect fitness positively that extend to quite a long way into the life of these animals. So rather than what we might intuitively expect, which is that something that has a beneficial effect at time t has a beneficial effect at time t plus 1 and plus 2, but diminishing, plus 3, diminishing. That's what we sort of predict when we think about bands of pleiotropy. They're tight, they're short, okay? What we're seeing is something more like this. Selection early in life is conferring survivorship benefits to remarkably later ages. And so what we're suggesting is that rather than coming to a mutation selection balance in these populations at a relatively young age, we're finding that balance way out here, sort of in the middle-aged fruit fly, if you like. Okay, so that's quite interesting. It also suggests that a lot of this work that has supported antagonistic pleiotropy and mutation accumulation has actually been documenting changes that are really late in the life of the animal. Moving around their longevity, moving around their fertility, but doing so affecting ages that are quite late and don't really explain why we see such great longevity and such robustness early in life. Okay, so spontaneous mutations affecting longevity accumulated rapidly in these populations. These were not recessive mutations. All of the animals tested were wild type in the heterozygous state. So these are actual mutations which would be affecting these populations from generation to generation in lab culture, and they should be actively selected against. So selection for early life fitness is helping to confer considerable longevity despite this relentless selection. So we think that this might have something to do with condition dependence creating broad bands of pleiotropy, the animal essentially being overbuilt for its early life mission and extensive benefits to that in terms of survivorship to later ages. So we suggest that the evolutionary theory of aging, with my cricket chime, needs to accommodate positive pleiotropy as a feature when we try to predict the longevity of organisms under particular selection regimes. We're not suggesting we can explain exactly why some organisms have a genetic architecture that allows the evolution of such things as similarity, or why others have extended post-reproductive longevity like ourselves. That's obviously constraints on the genetic architecture. What we're suggesting here, though, is at least in this one model system, we have another factor that we have to take into account when we think about the evolution of aging. So thanks very much. I'll leave you with some acknowledgments to all the people that helped make this happen, including, of course, Chris Kimber, the lead author on this paper. Thank you.